of Science, PhD. Our keynote speaker from Smithsonian National Museum of Natural History, Mr. Daniel Lumban-Tobing, PhD. Our humble moderator, Mem Kadek Dita Cahyani, Bachelor of Science, Master of Science, PhD. Lecturers, friends, students, all of us here. Good morning to us all. My name is Gabriela Trida Kurniatami. I am the student of Biology Department 2019. I will be your humble MC today. First of all, before we started, today's event is about International Thematic Lecture Series 4, Introduction to Systematic Ichthyology. And before we all started, we have to sing all together for national anthem of Indonesia. If may, Pak Sugi. Thank you. Thank you, Pak Sugi. Before we're about to start our event and discussion, let's all give our warm welcome to Professor Sabto for the greetings and welcoming remark. If you may, Prof. Sabto.
Okay, thank you very much, Gabriela, KB, for the nice introduction. Good morning, all of the students. Yeah, so very welcome to Bapak Daniel Lumban-Tobing, PhD. Yeah, you are still very young, and it's actually he is actually in Washington DC, right? So I don't know what time is it now there. It could be at 8 p.m. right? It's about
now it's 9 o'clock here.
9 o'clock. Okay. Hopefully you're not sleepy yet.
At 9.
I'm sorry.
Oh.
Yeah. So he is now in Smithsonian National Museum of Natural History. I think it's a very famous, I think, and as far as I'm concerned, they collect thousands of the specimen. Yeah, I think about I search here in the internet, it's about 147 million specimens of and artifacts. Yeah, specimens. Yeah. So that's huge specimens. I think last time I was lucky enough to visit Tepapa, New Zealand Museum, and also collect a lot of specimen there. But I think this one is bigger than the Tepapa Museum in New Zealand. And when the setup museum established, like I mean, like in your museum here, I think it will be very interested and very interesting for the visitors to come, and lots of things that we can learn in the past there, and then provide a lot of specimen to know, and student must know that this part of the fort, yeah, I think this one is the fort Stadium General, provided to you to build your competence, and we hope that we will having another and another uh, uh, scientific discussion, and also Stadium General, yeah, and or international thematic lecture, whatever it's said, but it's all about serving the students to build their competence. So we are lucky to have Pak Daniel Lumbantobing, yeah. Even although they can speak Indonesia very well, karena dia dari Indonesia, but for this time, please Pak Daniel speak in English, because we just make just to give the a sense of international for the students, and students will get familiar with it. So hopefully there's a lot of things that Pak Daniel will explain to the students and share the experience, how the systematic ichthyology is a part of very important things about biology. And then I searched also in the internet here that the collection of the National Museum is about 6 million specimens, yeah, in the world largest collection in North America, Neotropical, Caribbean, and West Pacific. Yeah. So I don't know if in Asia also available specimen there, especially for Asian fish collection. I'm not sure. Let Pak Daniel will explain that. And but last but not least, I expect that Pak Daniel will also give the students information regarding, hopefully, some of the students interested to come over there. Yeah, I don't know how to come to there. Maybe Pak Daniel will explain about the internship program or volunteer program that available there. Then students can come over there, and hopefully yeah, one of two students will lucky enough to go there. I don't know the way to do that. That's a Pak Daniel responsibility to attract the students there, huh? Okay, because I think the more students come to overseas, I think they will build their confidence, the more confidence. I remember last time when I was in Harvard University in Boston, and I heard that all the alumni of 15 students that get involved in Harvard National Model United Nation, it's already, they're very confident, and now they work in some perusahaan asing, ya, or the PMA, ya, and some also working now in overseas. So this is the big influence when the students can have a chance to go abroad. Like here, the moderator, I see Gaby, dulu begitu lugunya, ya, kelihatan agak culun. Sekarang betapa confidentnya dia sekarang, ya, Gaby. You look more confident, more beautiful. But come back from mana? Latvia, ya? Latvia, Gaby? Yeah. She come from Latvia. Back, yeah. It's about 6 months, or it's about 6 months. So that's the influence of the international, experience. Then I hope Pak Daniel also give opportunity to the students as well. Yeah, I don't know. Is there any or what we call invitation from the Smithsonian National Museum of Natural History or not, and explain the way to go there? Yeah, maybe we can mungkin kalau untuk urusan sangu nanti kita siapkan sangunya, minimal traveling from Semarang to Jakarta ya, Bu Dita ya. Iya. Saya kira itu dari saya sangat antusias. Terima kasih banyak ya. Thanks a lot for Pak Daniel who will be sharing soon ya the experience there, and give some thematic lecture to the students. And I just remind the students that you have obligation to summarize what Pak Daniel said in maybe 2 pages, Pudita. Yeah, it's about 2 pages about what Daniel said. Individual and collect too, because Pak Daniel, I am now a lecturer with the Budita for the subject of biodiversity, animal biodiversity, Budita. So the student here involved in they are a part of the students of having subject of biodiversity of animal biodiversity. So that's I can say that, and I finish it now. Thank you very much, and back to Mbak KP. Okay, Mbak KP. Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. Sabto, for the motivational speech. I think it was encouraging for me and all of the students here to actually go abroad. And before I'm moving on to the next activities, I believe we have to do presence or absency, which later will be put in the link in the chat. So please don't forget to do absency. Okay. For the next activity, I will pass on this opportunity to Ma'am Dita as our moderator. If you may, Miss Ma'am Dita.
Thank you, Gaby. Thank you so much. Okay, so today, good morning, everybody. Assalamualaikum warahmatullahi wabarakatuh. For everybody who is fasting right now, I hope you're still in the good stamina. It's still morning. So, and just as Pak Sabto said that you have a little bit a summary to be done, I'll let you know later. I'll send you the text to the WhatsApp group. Today we have international speaker. He's actually my good friend of mine. I never forget our date in the zoo, Daniel, in the DC. It's really nice though. Daniel is actually, he's an Indonesian. I'm just like, I will reading the CV of him just, through and through. And he's now a postdoctoral fellow in the museum, Smithsonian National Museum of Natural History in Washington, D.C. Daniel graduated from UI, biology department, Universitas Indonesia. And then he's an expert on taxonomy of fish. He's actually working a lot with Rasbora. I will let him tell you a story about this later on. Daniel, thank you so much to be here. I know it's very, very late at the night and you have a ton of work at the lab. But the thing is, I just missed that you were in Indonesia last January, I think, January or February. I just realized when I saw your Instagram and I'm like, oh no, I should invite you to Semarang. But I'm sure Prof. Sabto wants to invite you to Semarang because I really want you to be back to Indonesia.
You're really welcome.
Next
You're
time,
really welcome,
then.
Pak Daniel. Yeah.
If you want to back and teaching in Indonesia, Daniel, Undip, I think will be so happy to have you here.
Sounds good.
I don't want to read his CV a lot. You can find him. Just type Daniel Umban Tobing and then Ras Bora, and then you can see all the papers that he wrote. So just don't worry about that. So I will not reading all of your CV. Daniel, again, thank you so much. And I think I will not saying a lot about this. I think the time is yours, and please you can start the presentation. Thank you.
All right. Thank you so much, Pak Sabto, Budita, dan juga Gabriela for the introduction. Let me try to share my screen first. Right.
Okay.
All right. Can everybody see my slides?
Yes.
Yes.
Okay, perfect. Let me... All right. Good morning, everybody, and thank you so much for coming. Especially, I'm so grateful to Dita for the invitation. It's always a pleasure for me, and it's such an honor to share what I've been doing with my fishes and my research. And I hope that I can share my knowledge and I can encourage more people to study fishes instead of discouraging it, because I don't know, I hope it's not going to be too boring. But just bear with me, and I hope you can learn something about fishes today. All right. So today I want to talk about introduction to systematic ichthyology. So I'm currently a postdoctoral fellow in Smithsonian National Museum of Natural History in Washington, D.C. So I arrived here in, I think, 2020, February, just 1 month before the lockdown of pandemic. So it was quite sad, but we managed to survive. Here, my current project is about the systematic of rice fishes. The family is called Adrenichtidae. It is interesting because this group of fishes, how to say it, like half of the diversity of this group actually reside in the island of Sulawesi. So there are about 35 species in total globally, but half of the species are actually live in Sulawesi, and each species actually also show endemism. So they are endemic in like certain part of Sulawesi, or like a certain river drainage within a small part of Sulawesi. So it's a good system to be used to figure out the biogeography of Sulawesi. All right. So you've seen a lot of pictures of fishes. So these basically are photos that I took during the field trip and as well as during my research. Oops. Yeah, so what is systematic ichthyology? So in this talk, I want to try to, how to say, to convey some messages, to share about what I know about this field, because I've been doing this since probably like when I started my grad school in 2005. So I will explain about what systematic ichthyology is and why we need to study fishes. And also I hope I can show you the diversity of fishes just like in a glance. Although, how to say it, like, given the slides that I've just made, I don't think I cover like a tiny portion of the diversity of fishes. But at least I hope that you can see what we can do and why and how to study fishes. So that's another objective that I want to achieve here now. Okay. So etymologically speaking, ichthyology derived from Greek words ichthus and logia. You probably know the meaning of logia, which means to study. Just like the other branches in zoology, you see there is a herpetology and ornithology, mammalogy, for example, and the last I give for the examples is carcinology. So basically, all this field is we can call it like a minor branches in zoology. So we basically focus on certain group of animal, and then we really focus on that group to study any, how to say it, not any kind, but like most of the biological aspect of that group. So just like in these names, basically the identity of this field start shown by the first syllables of the name of the field. Like herpetology, people who do herpetology probably know they study amphibian and reptile. Ornithology, they study birds. And of course, mammalogy, you know, you might, you are likely know to know what they study. And probably on the right side, these are fields that not, how to say, because the group that they cover are relatively not small, but actually very diverse. But in terms of the number of people that study, is not as many as the one on the left, because of course we are mammals, so very mammal centric in terms of the number. But like entomology is the field that study insects, arachnology, spiders, malacology is mollusk, and carcinology is the field that studies crustacea like shrimp, crabs, and copepods, for example. So then what is ichthyology? So the first two syllables is ichthus. What ichthus actually mean? It's from the Greek. Of course, that means fish, because I've been showing you all the photos of fish. But then the $1 million question is what define a fish? What characteristic that can be used to define this animal? Here I give you an example of common, probably something that, how to say, it's like layman people can notice that this is pretty much like what typical fish can look like. So the first characteristic of fish, they have a backbone, or we call it vertebrae in our scientific terminology. That's why they are called, they belong to the group called vertebrates. How do we know it? Because we basically do observation in their internal morphology. So here I give you an example of a minnow that I collected in Southeast Kalimantan, or Kalimantan Selatan. I was excited to catch this fish because I've never, at that time, I've never seen this minnow before. And then I managed to do X-ray. So at the bottom is basically when you do X-ray photography, you can get information of the bone, roughly speaking. So there are many characters that, not many, but some important characters in fishes that can be used to diagnose them. One of them is the number of the backbone. So as you can see in the middle part of the body, that's what we call backbone, or in Indonesia, pretty much we call it tulang punggung. Similar to all mammals or all other vertebrate, from fish to bird, amphibian, all they have this a series of bones that support the body in the axial structure. So fish do have it. So that's one of the character. That's why we call it vertebrate. They belong to vertebrata. And here on the top with the white background, I give you an example of cleared and stained specimens. So these fish on the top also belong to the same family, which is cyprinids. But they how to say it, like they seems to be like closely related. So when it comes to resolution, I like examining the cleared and stained specimen like on the top, because you got more information, and of course if you can dissect these specimens, you can get much more information, and even in three-dimensional fashion. And that's something that can be used in our study, especially for the systematics of this group. And then the second, of course, pretty much, how to say it, like fish literally live in water. Everybody knows that. And they have fins, so they move around in their habitats using their fins. So here I show you different fishes, but this is Indo-Pacific tarpon. So in Indonesia, we call it ikan bulan-bulan. So usually they live in like the coastal area, even like they get to the estuarine part of the river, like mouth of the river. And this one I collected in Sulawesi, in central Sulawesi. So this is this fish belong to quite, how to say, primitive group of fishes. So they've been in this world for like probably like 100 million years ago. So this is quite in terms of evolutionary time scale, this group is basal. So basically, if you want to study fishes, this is a good model because it gives you like not typical kind of fish body, but at least like they still have all the primitive characters of fishes. So before we go further, I would like to give you, I would like to explain you about the terminology that we use to give you the orientation of the body relative to each other parts of the body. So in, I mean, this apply to all field of zoology, not only for ichthyology. So dorsal, usually like the part that top of the backbone. Ventral is towards the, we call it the stomach part, and anterior is how to say it, like towards the head, while the posterior is the opposite of anterior. So that's towards the tail. And this photo, usually we use this kind of setting, and we call it lateral view. In ichthyology, we usually use the left side of the fish. So if you want to take the tissue sample for molecular study, try to get the right side, because usually in for fishes, we use the left side to how to say, like, to be like the standard in terms of photography, taking pictures of these individuals. Yeah, so this is the lateral view, the left side of the fish. So in describing a different part of the body, we use this terminology: dorsal, anterior, ventral, and posterior. For example, the head is anterior to the body, and the body is posterior to the head. So this is like the scientific terminology that we commonly use in zoology. So we don't, I mean, in terms of scientific, how to say, papers, for example, we don't say in front of, because it's sometimes we don't know what we are referring to, because it can be very relative when we say in front of. So these are the terminology that we use to orient different parts of the body. Okay, probably most of you already know different types of fins in fishes. So the dorsal fin on the top, like dorsal to the backbone. It's usually in like primitive group of fishes, they only have like one single dorsal fin. But in the modern group, like modern lineages of fishes, they can have like two dorsal fin, the anterior and the posterior one. And then the most anterior fins is pectoral fins. Notice that we use plural word, fins, because they always come in pair. Unlike the one in dorsal fin, they only have like one. And that also apply to pelvic fins, also always come in pair. And then posterior to pelvic fins, usually we have anal fin. So why we call it anal fin? Because usually the opening of the anus is just anterior to the anal fin origin. And the most posterior fin is called caudal fin or tail. Typically, fishes will have two lobes, anterior and dorsal lobe of caudal fin and ventral one, just like in this example. So we have dorsal and ventral lobe, but it's not in pair, because it basically like one single fin. It's just because it has like this deep convex part, so it looks like it has like two lobes. I mean, like, clearly there are two lobes. Fishes are so diverse, and fins also one of the diverse expression of in fishes. I mean, here the example that I've shown you is the primitive lineage of fish. The primitive fish usually have only one dorsal fin, and then you notice the pectoral fins always in pair, but the position is relatively ventral. This is typical in primitive fishes, like in how to say it? Example of primitive fish is like, for example, arowana. also like cypriniforms like loaches, ikan mas, ikan wader. These are examples of relatively primitive lineages of fishes. And you can always tell that their pectoral fins is very ventral relative to the other how to say it, modern lineages of fishes. And on the right side, this is an example of modern lineages of fish. They are more, how to say it, they show many more derived characters. For example, like this, the dorsal fins in more derived fishes usually have two parts. The anterior dorsal fin, they usually have spine, and the posterior part of the dorsal fin, the rays usually built by the softer rays, so it's not spinous. And then what else? Oh, so here probably it's not too clear, but as you can see, I'm trying to emphasize on the pectoral fin of this fish. So as you can compare here, this one, the position is relatively dorsal. This is the character of derived fishes, more modern, the modern lineages of fishes. And I give you different arrows to show the difference. So here there is only one dorsal fin, but in this silver side there are two parts of the dorsal fin. And another example of primitive fish is eel. Kalau di Indonesia bukan bisa dibilang belut sih, tapi kalau di Sulawesi disebutnya masapi. And you can tell even they have a very, what to say, even though they are primitive, they belong to like basal lineage of fish. They have very modified fin structures, because as you can see here, they have like very elongated dorsal fin that connects to the caudal fin, and even further to the ventrally, it also somewhat merged with the ventral fin. So it looks like a continuous series of fin. And what is interesting, eel usually don't have pelvic fins, because they use their body more instead of their fin to move around. So they pretty much not similar, but somewhat similar to snake in the terrestrial habitat. So they rely on their body to move around. Okay. So I hope this example can give you a picture about how diverse fishes can be. So these two fish are primitive, while here Telmaterina opudi, endemic in Lake Matano in Sulawesi, they show more derived characteristic of fishes. And here also a good example of modern lineage of fish, although you cannot tell whether it has like two dorsal fin or not, but actually the anterior part of the dorsal fin comprised of spine. So the fin rays on the anterior-most part of the fin are spine, while as you can see here further posterior, you can tell that these fin rays are softer, relatively softer. You can see it actually branching and not pointed. So you don't have to worry if you catch this fish in this part, but you really have to worry if you hold this fish in the anterior part of the dorsal fin, because this is pretty much like their how to say it, like mekanisme untuk apa melawan misalnya the predator gitu. They also have like spine in their anal fin series, like in the anterior part of the anal fin. So, and then also notice here the pectoral fin, although it's not too clear, but you can tell that it is relatively more dorsal. So whenever you see this characteristic, you can always tell that these fish are relatively modern. I mean, compared to like eels or like that tarpon that I've shown you, this is relatively more recent evolving. So like probably after the Cretaceous era, while eels and tarpon, they already evolved like probably like 150 million years ago. They already exist. Their lineage already exists then. Okay, sorry. So we know fish have backbone. They live in water. They have fins. They move around with fins. Although the fins can be very modified in some lineages, because depending on what they the way they live in their habitat, just like eels, they have completely different way to move around compared to like typical fish like the one I show you here. And the other characteristic is that they are cold-blooded, ectotherm. So they cannot maintain the temperature in their body, unlike us. Like we are mammals, so we can physiologically maintain our body temperature like in certain range, but for fishes, almost all of them are ectotherm. Although some fishes like tuna, for example, they can create heat in their body in certain part of their region, but still they are not entirely endotherm, which is the opposite of ectotherm. So ectotherm is called cold-blooded. Endotherm is like warm-blooded, just like mammals or birds. And the other important characters to define fish is that they breathe using their gills. So they obtain oxygen via gill structure. So I will show you. I think gills structure is the coolest part of the fish, because there are so many bones that constructs this structure, and there are so many. It's like the source of how to say it, like so many information that can be used to diagnose a different group of fishes, depending on how they eat, how they even like the way they swim, that affect the morphology of the gill. So here I'm trying to show you. This is a stained specimen. So what you see here are red structure. So those are basically the bone, because we use alizarin red. So that kind of dye will stain all the bone structure to look red, while the blue are usually the cartilage. So we use different stain we call it Alcyon blue, and it will stain the cartilaginous structure. So it's a good way to prepare your specimen if you want to study more in details about their bone structure, and we call it osteology. And we use these kind of methodology if we want to study or to have like more detailed pictures about the systematic of the group, or how to say, like more like the classification in a more evolutionary kind of thinking, because fishes have much more bone than other vertebrate, given their gill structure, and also given their oral structure that they use to breathe and to eat. So here is the opercle, so the bone that covering the gill part. So the gill part is here. So anterior, which is to the left, to the left. Of course, you notice eye and then the mouth, and it's interesting how this is a endemic genus. Sorry, this is a genus that is endemic in Lake Poso in Central Sulawesi. Very cool fishes because they got quite big, while their relatives in the same family, they got only like probably like 2 centimeter in standard length, but they can be as long as like 20 centimeters, and they are can only be found in Lake Poso. In order to see the internal part, you need to dissect the opercle, this part. So I did a lot of dissection with this kind of specimens. So here I already took the cheek part of the fish, and you can see this part is basically the gill, and you can notice these are the gill filaments where the, how to say, the respiration of fishes take place in this part. So there will be like blood capillary through this each of the filaments, and there where actually the how to say, oxygen and carbon dioxide get replaced. I don't want to get too into detail, but basically you can see here, this is the gill part. And here I show you different kind of preparation of the specimens. So previously you still see the muscle, which has that whitish coloration. So here I use mostly muscles, bones in red and cartilage in blue. All the whitish muscle the whitish part are the muscles. But in this case, in these specimens, we cleared the muscle. We cleared most of the soft tissues. So example of most soft tissues are muscles. So what you can see here are pretty much the bones and the cartilage. And I don't know, I see a beautiful view here, because what I see is so many characters and so many weird stuff going on, because basically how to say, this group of fishes, they have a highly evolved part of the gills, because they are minnows that are found in Sundaland. So you can find this Pectenocypris in Sumatra, Borneo, but not in Java. And they are pretty much like phytoplankton. So they eat all these tiny algae and they sieve all these tiny algae using their gill rakers here. So you notice that it has a very how to say it, like very dense and tiny arrangement of gill rakers. Usually, the number of gill rakers can tell what kind of food the fish eat, because this group of fish have relatively numerous. I mean, like so many. This is probably, there are probably like hundreds of gill rakers compared to, for example, like the other minnow only have like a dozen, which are like 12 gill rakers, and they now, as you can see, have so many. Okay, I can talk so much about fishes. So here I tried to show you what actually the gill structure is, because if you compare, the skull is still there, and the mouth part, the cheek part also still there. But now I highlight only the gill structure. We usually call this as a gill basket because it looks like a basket. And we use these structures many times in our study for, especially for figuring out the evolutionary relationship, I mean, compared to the other fishes, because there are so many information that you can get from this structure. I mean, compared to, for example, like mammals pretty much, they don't have gill, so they lack this cool part of the head, so they pretty much don't have so much information that we can get in a fish. So I hope I can give you why this structure is the coolest part of the head. So this is the dorsal view. So if you see from the top of the head and looking down, this is what we call gill basket. So here I dissect the upper part of the gill basket, and then this actually, the opposite of this part. So the other side. So this is like the right side, while this is still intact is the left part of the upper gill basket. And in this group of fishes, which we call cypriniforms, the how to say, the back of the gill basket modified to become a pharyngeal teeth. So they don't have teeth in their oral, in their mouth, but they use this part to bite, pretty much bite and chew, unlike other fishes that can use the teeth in their mouth. Given that this group don't have that teeth structure in the mouth, they lack that kind of teeth, so they only use this part to chew their food, which is pretty cool. All right, so we got 5 characteristics that define a fish. So my apologies, because some of my slides still in Indonesian. I mean, I've given this kind of talk like completely in our language, so and I didn't have the chance to translate it. So based on those 5 characteristics, fish are a group of vertebrate that live in the water. They swim using their fins. They are cold-blooded, and they breathe using their gill. This is in zoology what we use to define fishes. But what is interesting, using that 5 characteristic, so these 5 ones, we have this phylogenetic tree, and which lineages that show those characteristics. only 3 of these that actually show the true definition of fishes based on those 5 diagnostic character: lamprey, sharks, and teleos. Shark belongs to what we commonly is refer to cartilaginous fishes. Teleos is the modern, how to say that? Teleos is relatively modern, but that also include the tarpon and the eel that I've shown you. And lamprey is what we call as ikan tidak berahang or agnatha. So they don't have a jaw, but I don't know, probably you are familiar with this kind of diagram, which is a phylogenetic tree. So tetrapod and teleos, they share the same common ancestor. But this kind of grouping, we call it polyphyletic because one of the lineage that share the same ancestor are not included, they are excluded. So this is not natural according to especially the philosophy of cladistic, because what is natural is only monophyletic group. What is monophyletic group? The group that share the same common ancestor. So in this case, teleos and tetrapod, because they branch from the same split or common ancestor, they can be called a monophyletic group. But in this case, using the characteristic of fishes, apparently tetrapod are excluded because of course they don't live in water, they don't have fins, they don't have gills, they lack that all major diagnostic of fishes. So by that definition, if we want to classify a natural group, that tetrapods should be included. But then that would make the definition of fishes is like, we are pretty much all fishes. So tetrapods are pretty much like fishes that live in terrestrial habitat. They just, how to say it? They just abandon all the fishes characteristic. But again, how to say, the pragmatic level, because we don't want to study all vertebrate, because I mean, like ichthyologists are very focused and interested on fishes. So we just use the definition that, how to say it, those definition that defined by those aforementioned 5 characteristics. Anyway, but if we want to say naturally, all the vertebrate are fishes because we share common ancestor, even with lancelet, even with hagfish. Okay. Yeah, another slides in Indonesia. So what is ichthyology then? Ichthyology is the branch in biology that study fishes in a scientific way with an emphasis on a number of subdisciplines which include into what we call as basic science. I will try to explain more about basic science in biology. I think this part, to know what basic science is and why, is really important because our major is biology, and biology is pretty much fundamental, or in other words, basic science. And to, let's say, to be able to properly study something, we need to really know what we are dealing with. So I hope I can deliver the message why knowing basic science compared to the other kind of science is important for us who have major in biology. Okay, so bear with me because this is gonna be quite, how to say it, tedious or too philosophical probably. But basic biology include taxonomy, systematics, or slash systematic. I will explain later again why we call it slash systematic. Anatomy, morphology, ecology, behavior, and also physiology. I give emphasis on basic science because I want to talk more about this. So basic science versus applied science. Why we need to know about this as a major of biology. So here, basic science, according to these slides. So in basic science, we study, what we study is something that we add to the library of knowledge. So this is like, how to say it, like we are not trying to solve something like problems in the real life, but we are adding more and more knowledge. When we know, we objectively and empirically have all the evidence to say that we know, but it doesn't have to be something that can solve the problems or issues in our real life. So this is important to know because what this is basically probably what we are going to do in the future, especially for a student, undergrad student who try to get a bachelor degree in basic science. So they gave example like to study whether ants have knees, whether certain group of fishes have gills or lack gills, whether why salamander still retain their gills in certain stage of their life. And this is not something that can solve our daily serious problem in this world, but we basically add more knowledge. Basically, we know more than before, but we are not trying to solve something. But in the contrary to applied science, we study something, our subject that we study, our objective is to solve problems, especially something that we deal probably on daily basis or probably something that we encounter if we do something, but the objective is different. But then how to say it, it sounds like something is more important than the other, but actually not, because in basic science it's more like when you add more knowledge in the how to say it, in in humanity, we actually saving something for the long term. So we probably, I mean, our new knowledge probably is not going to be like useful like the next day, but at some point in the long term or probably soon, we don't know, probably we know, but it doesn't matter. But I would say like the important thing is that there's not, it's going to be useful in the future. While applied science, you'll be doing something that can solve like immediate issues or problems, probably on a daily basis. And that's why, how to say it, like there are consequences, and that's something that I've been dealing. And you meet like different people that work in applied science that probably make more money than people that do basic science. But how to say it, like the thing with basic science is pretty much like exploring the unknown world. So if you are passionate about finding something new, you are good at this kind of field. I mean, you're gonna be someone that go everywhere in this kind of field. That's why one of my supervisor in my undergraduate always says that when you do basic science, you need to be very passionate. Otherwise, it's not gonna be like something that you really want to do. So how to say it? It's not like I'm discouraging you, but it's just like showing you the reality in our field. Of course, in biology, if you have like major in biology, you still can be applic you can still do something that are applied. But how to say, in the real life, probably you're gonna deal more that towards to basic science. But of course, it's not like how to say it, like black and white. They're always like overlapping, and even like these are like complementing kind of field. There's no like sometimes like splitting hair kind of thing. But I think I think it is good to know the difference between these, so we know what we are doing and we know what's the consequences are. Okay, blabbering. Okay, so here I give you example. So like basic science, physics, and then the applied of physics would be engineering. For example, you can build a house, you can solve the problem of housing, even like you can figure out uh, the daily problems in figuring out how to get a comfortable house. And for chemistry, like I don't know, industrial chemistry. And in biology, it's not always like basic, because people that have major in biology can also pursue something in medical field. And even a lot of my friends now become conservationists. That's pretty much like predominantly the applied part of ecology. You will be dealing with more people, and you will figure out how to uh, keep ecosystem sustainable. And of course, I've seen a lot of students majoring biology and then got their bachelor and then they become like conservationists, and that's very applicable in Indonesia, especially given we are like one of the most biodiverse, one of the most country with high biodiversity. And on entomology, I've seen like curators in Smithsonian in my museum that also work in trying to control the pest. And now, what is the applied science of ichthyology? Yeah, so fisheries are we usually, sorry, or we call it perikanan. So perikanan is not the same as ichthyology, because your main objective is to breed or harvest fishes. So you try to create like controlled environment in order to get more harvest of fishes. And that's not basic, that's like applied. Of course, to be able to do that, you need something, you need like a lot of knowledge from ichthyology. Without that, in the absence of ichthyology, we cannot pursue fisheries for sure. Why study fishes? Yeah, the first 2 points, that's something that like economical utility of fishes. We know that a lot of people eat fishes still, billions of people. That's like probably one of the only protein source in certain part of the world, and people love eating fishes. I mean, not trying to be how to say it, like discourage the other fields in zoology, but not many people eat amphibian or reptiles, but a lot of people eat fishes. So that's something telling about its applicability. And other non-food economic values like people love beautiful fishes. They love pretty fishes. That's why they have pets, and that's like quite a highly valued trait nowadays. The the rest is like it's more like the biological aspect of fishes, because fishes are fishes are so diverse. There are still many species that undescribed, so it's still unknown to science. So if you become a fish taxonomist, you have a lot of homework to do, just like me. Sometimes it's just like too much, but then that's why we need more ichthyologists who do taxonomy and systematic. I hope I can encourage more people among you as well. And then it is they are a great system to shed light on many problems in fundamental biology, because why fishes are pretty much like the most basal group of vertebrates. So their evolution will tell you more about why we are now, why we evolve to become like human, and a lot of things going on in their genes, genetic structure that can tell something about our evolution, either directly or indirectly. But a lot of things, especially given now in when people study genomic, and also they are great models to solve many problems in applied biology. Yeah, it's pretty much the same. I mean, when I said applied biology, because you can probably solve like certain kind of disorder, like genetic disorder that can be this, how to say, like certain kind of genetic series in fishes that probably can shed light more about genetic disorder in mammals, because they are pretty much basal. So we need their evolutionary background in order to study more about more recent divergent, even though they live in different habitats. I mean, I can blabber more about, and give a lot of examples about it, but we are in the genomic era, so there are also many papers that can prove this point. Yeah, so that's basically why we study fishes. I can give you more reasons, but hopefully that can convince you that fishes are cool. I had another boring part. So yeah, basic biology include taxonomy. So I want to explain about what is taxonomy. So this is the branch in biology that classify the diversity of living organism. So without taxonomy, everything will be messed up. And then we need to group this living organism to different hierarchy, and that's we call taxa. That's why the name taxonomy derived. Given this objective of this field, so there are like several steps, like become the standard operation SOP, standard operational procedure in taxonomy. First, identification. You need to identify those living organisms, and then in order to be able to communicate, we need names. So it can be universal, and we call it nomenclature. And then those names need to be classified, of course, according to evolutionary information that we study. And of course, this description, like taxonomic description, which is important part and usually the final step. So what is taxa? So usually like kingdom, phylum, class, it is called rank. But what is called taxa is actually the real thing. I mean, like the living or the organism. So Animalia, Chordata, Mammalia are taxa, but kingdom, phylum is the rank. Can be confusing, but the way we call it taxonomy because we refer to the real thing, which is the organism, not the rank. And rank can be subjective, but taxa should be real or natural. Ideally, I mean, like ideally, we are trying to recognize natural group which are taxa. The first step in taxonomy is identification. So we need to get the identity, not only like local identity, but also identity that can be recognized globally and hopefully universally. That's why we have nomenclature, binomial nomenclature that hopefully everybody, not everybody, at least people can agree with relatively globally rather than being local. For example, I mentioned about Indo-Pacific tarpon. In Jakarta, we call it ikan bulan-bulan, but in other places, like in Sulawesi, something else, and that's not something that global. So that's why we need something scientific, more objective way to have identity of this group of organism. And what is important is they are recognized based on a series of attribute or characteristic that can be recognized or can be observed clearly. Usually we call it diagnostic, the diagnosis of certain taxa, a series of characteristics that is unique, that are unique in that group that can be used to identify them. So identification is something that how to say, imperative when you do study like field inventories or ichthyological surveys. I mean, like this is like typical, not only in ichthyology. If you do like inventory of amphibian in Sumba Island, and then you will collect all that you can get when it comes to amphibians. And then, of course, once you have all these amphibian taxa, you need to identify them. Just like here, these are some excerpts of photos that I took during my my field trips, different number of field trips to Sulawesi, different parts of Sulawesi. So it is important to know the identity of ideally each of your species that you have collected, and how to get to that point. We usually use either taxonomic description or identification keys, but usually for not quick energy, but more like to get to the generic level, the level of genus, we use general identification keys for certain groups. And then in order to get to the species level, then you need to go to the taxonomic description, usually of the species. So it can be tedious, So that's why in order to do that, you need to know what character are used, or at least what character are commonly used or typically used in that group, because the wording can be very verbose, even like the terminology can be foreign to you, but that's why the point that we have so many minor branches in zoology, because each branches try to give their best when it comes to identification. But of course, when you become like taxonomists, consequently, you need to provide the public a good diagnostic or a good character to diagnose different species or taxa. The one that easy to access, probably something more external. Ideally, you can use all the external morphology or anatomy. So layman people means that people that don't work on your group can access that part without having to destroy the specimens. I mean, for example, I won't use like the upper part of the gill basket as like the diagnostic of the species XXX, because it would be very, let's say it, very inefficient. I mean, it's not good for other people that don't study your fishes, because they need to open, they need to destroy basically the specimens. So that's why it become clear. I will explain later what is taxonomy and what is systematics. And when you deal with this field, you get the hang of it, why there is like differences in different level, basically in the way how you diagnose those different taxa, how you need to provide the public with a series of characteristics that relatively easy to be accessed. So yeah, here are example. So usually identification key, for example, like here, number 1, pelvic fins moderate to large, that's something accessible. And then you can measure the proportion using percentage. And then usually like a series of character that you can follow, and then you go to the next number and then find, and it getting like narrowed into, for this case, the genus. And I mean, of course, given that so many undescribed fishes in this world, you probably end up with completely new genus or completely new genera. And then that that's why it brings to the next steps, which is description process. So you try to, how to say it, like expose all the identity of a taxon with a detailed explanation about the characteristic and the condition, each of them, which can be recognized clearly. So that's why clearly means that they are accessible, they are easy to how to say it, easy to measure, can be recognized by most people. I mean, like for example, like number of fin rays is something that can be counted by everybody. While, for example, the proportion of the barbel compared to the head, while barbel can be destroyed by something that during the life of the fishes. So there are some bad examples of a diagnostic character, but I mean, the clearer and the more accessible the character, the better. Yeah, so I did several field trips to different part of Sundaland, which are Sumatra, Borneo, and Java, and I've encountered a number of undescribed species of rasbora. So here I'm not showing. I mean, all of these are not undescribed or unknown to science, but some of them, and some of them are still my homework. So yeah, something to be finished. So a lot of pressure when I see these photos. But this is pretty much like the living coloration of these fishes. Once they got preserved, once they died, once how to say it, even like in certain stage of their life, they might not be showing the same coloration. So, and actually this living coloration can be also informative to diagnose the species. So it's always good to record this kind of living coloration because you never know. I mean, the more character you have and the more steady the characters are, so the more the better you can provide to the public, especially when like people in Sumatra, they encountered like these species when they are alive. So they are familiar with the living coloration. So it's easy for them when they took photos when they are still alive. These fishes are still alive. So I mean, the more accessible, the more informative, the better your taxonomical studies. Yeah, so I described 4 new species from northern Sumatra, so that include Aceh, North Sumatra, and some part of West Sumatra. So I used color pattern a lot because all the other characteristic that have been used by previous ichthyologists are not informative. So what does it mean? It means that they overlap in among species. So for example, like the number of the scales, the lateral line scale, they are overlap. Their fin race number also overlap. But the only available accessible characteristic are the color pattern, and these are something new. I mean, previous author never used this, but then you need to describe this new series of characteristic, new series of diagnostic characteristic to the public. Of course, this can be tedious because you need to compare different specimens from different locality. Hopefully it represent different population. So it means that if it keeps showing the same color pattern, it means that it is steady and it's good to represent this species. And luckily, I managed to do genetic analysis, but after this publication, and apparently based on those molecular analysis, it support my hypothesis based on morphology. So yes, I mean, the more you got supports from different lineage of science, the better. It's I mean, new species are just hypotheses. It's not something that how to say, like steady. It can be falsified by other studies, so you need to be prepared for that. But whenever other authors or other studies confirm your hypothesis, that means that you did your homework well. hopefully you will be the one that that discover that hypothesis. But again, you cannot win everything by yourself. So that's why it always good to have collaboration, always good to involve local people, and of course, always good to acknowledge their effort in your work. Okay, so wow, I talk so much. How many? All right, so in identification and the description, those are two steps commonly used in commonly done in taxonomy. But you can here I highlighted what is important in those two works. So pretty much all about recognizing different characteristic, atribut yang dapat dikenali secara jelas. Secara jelas artinya bisa diakses semua orang, gampang dicapai, mudah dipahami dan sebagainya. Jadi jelas dalam artian relatif lebih bisa diulang oleh banyak orang, bahkan orang-orang yang bukan ahli di kelompok tersebut. Jadi itu kenapa, tapi dua itu jadi sorry. So in identification, in identifying a living organism, we are dealing with a characteristic, but we want to get the identity of the name. So that's why we try to to follow all this series of characteristics that can diagnose that taxa. But in the description, because we are finding something new, or we are not necessarily new taxa or undescribed one, but probably from the previous papers, they didn't really describe well. So you need to add more about the identity of that name. So in the description process is pretty much like not the opposite, but like you are going back. So you're trying to get the identity of the name. So you need to list all the characteristics that can be useful to give the identity of the name. And yeah, in systematic / taxonomy, we live to find new character, or we try to get character that distinguishing different taxa. That's basically what we do. So yeah, characters. Different types of characters. I've mentioned a lot of morphology because I predominantly do morphology in my study, and I also do molecular. So I use DNA mostly. I think all of my molecular analysis use DNA sequences, and some character can be cytogenetic, like chromosome number, for example. But that's really something probably that applicable to higher level of taxonomical rank. And then even like the behavior characteristic can also be informative, because like, for example, like mating behavior can be different. I mean, clearly different species can exhibit different behaviors, of course, because they pretty much have different couple, different pair that completely belong to different lineages, evolutionary speaking. That's why they have different differences in the behavior. That's why they weren't matched with other species, because basically they don't behave the same. And also ecological characteristic, for example, sometimes it can be secondary, but it can lead you to find something new, like new species. For example, like in freshwater fishes, especially fishes that live in the upstream part, they tend to be really isolated with other that live in different river drainage. For example, fishes like Rasbora in Tasikmalaya, like region, and all the river that drains to Indian Ocean to the south. I found that they are actually different from the one that live in the river drainages that drain to the north because of the mountain range, because ada pegunungan yang membatasi, yang mengisolasi dua populasi besar. That become like a very effective geographical barrier. So when you how to say, whenever you see like certain mountain range that shape like U, and then like in the lower part of that drainages, they don't share with the next neighboring rivers. Usually the upstream fishes are different. I mean, like they are like completely endemic to that drainages. This is true in Borneo. Like if you go to Meratus Mountain and then you try to collect loaches. Loaches is like upstream kind of cypriniforms from the western part of the mountain, and then you did collecting in the eastern part, and somehow you find that they are different. I mean, like externally, like clearly they are different, just because they never like for a long time they've been isolated from each other, although they are like quite close. But that mountain become like barrier for the genetic for the gene flow, and that always the case for upstream lineages of freshwater fishes. And that's why I could describe like these 4 new species of Rasbora, because this lineage of Rasbora, they only found in the upstream part of river drainages. You won't find these lineages in this particular lineage in like middle part of the river. I mean, when you see the river lack all the big rocks and boulders, and they look more like with the slower rapids, and look more like yeah, how to say, like warnanya kopi susu. That's like already you are in the middle reach of the river, and you won't find this kind of lineage. And if you go further upstream, when the water are rapid with the rapid currents, and then they look more clear, you will find these packs of fishes. Yeah, so that's something from the ecological aspect, and you can kind of like figure out, especially if you are interested in finding something new. Then good thing about freshwater fishes, go up go up to the stream. But that actually not only applicable to, I mean, similar thing happen in different habitats of water, like aquatic habitats. For example, like in coral reefs, probably like certain coral reefs are isolated from the other, because just because they are ocean currents that become like effective barrier for those two, or more than two. Anyway, okay, so
Daniel, I'm so sorry. Can we wrap up in the 2
okay,
minutes maybe, because we have a couple of questions? Thank you. Sorry.
okay. So sorry. Yeah, so morphological character, morphometric is basically measuring. So this is quantitative kind of characters. It can overlap and it's not discrete. Meristic is a kind of a discrete character and you can count. And a lot of characteristic in fishes that can be used to diagnose different taxa. Color pattern are my favorite because at the end of the day, I only found these are good characters, and it got supported by molecular. So this is example from my previous works. You can tell, but only after preservation and when they died, the color pattern become apparent and quite distinct, but they are steady and they are, how to say it? Yeah, they are congruent in that population. That's why I'm confident to use them as a diagnostic character. Internal anatomy, but then you become someone that probably everybody will hate you because you're using less accessible kind of characters. Usually we use this in systematics. Molecular can support morphology and the other way around. I use DNA sequences a lot. Oh yeah, so this is the important part. What's the difference between taxonomy and systematic? So in systematic, it's pretty much like taxonomy plus a phylogenetic tree. So in systematics, we use the evolutionary framework more because we try to identify monophyletic group that reconstructed using the phylogenetic methods, and we will describe that group and give the names to that group. That's the main objective. Yeah, so it's a evolutionary relationship among different taxa. We try to figure out the sister of the other. Okay, so that's pretty much. So in systematics, we can use a lot of detailed characteristics. So for example, here, this is the work that I use. I use a very detailed characteristic using bone. I mean, I won't use this for taxonomy because it's not accessible for public. I mean, like layman people won't be able to get this. But it can be used in reconstructing phylogenetic tree, finding like synapomorphy that support that group. So here, you need to dissect the bones and really do like the staining. Like here, example I'm showing you, different parts that are useful to diagnose different group, and you can see differences, even though this is something like complex, but I hope I have more time. But again, this is the talk that I gave last year in our meeting. So basically, I'm trying to describe different part of the structure using muscles and also bones. So yeah, I'm pointing at different differences. And I won't use this in taxonomy. I just use this for systematics. I hope I can, you already get my point why I use this, not for taxonomy, but now. So synapomorphy is the one that become evidence in defining a monophyletic group. It's shared by the same lineage, given the same common ancestor. So these are the lists that I gave as the evidence to support, in this case, Beloniforms, an order that I now I'm focusing. It's just another discovery. I mean, just like finding new species, new characters is just finding new characters feels the same. I mean, you basically exploring different biological entities. I mean, like when you go to the field, you explore the geography, but when it comes to morphology, you're exploring the whole fish. I mean, like, yeah, you get into detail with the fishes, but basically it's just a matter of exploring. So you are pretty much Dora the Explorer if you become systematics or taxonomists. And if you are passionate about finding something new, then this is the field that you want to pursue in your life. So the question, what define Ichthyology? So yeah, I found also different characteristic using the same methodology, using muscles and bones. Yeah, this is more in the family level. All right. So that's pretty much what my talk. Sorry if I'm too boring, but I hope I'm not discouraging you, but instead more like encouraging you to study fishes. And I hope someone among you will become an ichthyologist. Thank you.
Thank you, Daniel, for the presentation. I know that some of the students also thought that I'm a boring, lecturer in front of the class because I said exactly like you said. We are doing a basic science, so we are not trying to solve problems. But I love what you say, that we are researching to add the library of knowledge, and we are exploring the unknown world. So that's, I think, basically what we do as a biologist. So it doesn't have to be everything is applied science. There's a couple of questions though. So some of the question I think you already answer. However, I also want to ask it again. So the first is that how many species that you have described so far? And the follow-up question from that is that, do you have specific taxa as a focus, or are you a generalist? That's first. Can you maybe share a bit about that?
All right, thank you for the question. So yeah, so far I've described, I think, 10 species.
Oh,
Yeah.
wow. Okay.
Yeah. Eight species of Rasbora, all from northern Sumatra, and 2 species of rice fishes from southeast Sulawesi. Yeah, I mean, I'm now writing up more description, but then how to say it, sometimes when it comes to systematic, like doing the taxonomy part and also the systematic part can be too much. So in my defense, and then there are like lab works, you have to say, like it can be very hard to focus on, like, for example, like counting the scales and then pipetting. Like you basically are doing different things at this pretty much the same range of time. But I think when when it comes to systematic, my supervisor always say it's become like your lifetime project. So yeah, it's gonna be your homework the entire life.
So do you have a specific taxa? Do you will go into this Rasbora like the minnows, or are you also study the other group of fish?
Yeah, so I've been focusing on Rasbora for my grad school, but for my post doctoral, especially the one here, I've been doing rice fishes, especially the ones from Sulawesi. And yeah, we collected quite well, a few undescribed ones from Sulawesi, and I think there are more, but sadly, because of the nickel mining in the eastern part.
Oh, right. Yes.
The last time my friend went there, and he couldn't find any fishes in that part. And even if you do Google Earth in that part, you can see how destroyed, how those regions become devastated by the coal mining. Yeah, it's very sad. I mean, such a high endemic, such a region with high endemism, with many unknown species, but they're already extinct probably at this point, and it's so devastating.
Yeah, that's true though. I actually want to ask that question, but I think my question will be the last one. So if any of you want to ask a direct question, just raise your hand and I will give you the opportunity. But I will ask you the list that I have. So the second question is, how long do you spend to describe one species? You know, in
Good question.
yeah.
Yeah.
How long does it take?
It can vary, I think. Sometimes if you find something very different, very distinct, and it's like understudied kind of group, and they are quite relatively small group. I mean, for example, the genus probably only have like 3 species. Then your homework is gonna be like shorter than describing like a genus that already have like 150 species, because then you need to compare more specimens, more species. Yeah. But good thing about doing inventories in like isolated regions, then fewer species been described from that area. So you can kind of like focus on those species that been described only locally, because usually in different region, it would be completely different assemblage of species. For example, if I collect now in before the nickel mining in East Sulawesi, I probably found like 2 new undescribed species, and then I don't have to do a lot of comparison, like species comparison, because barely been described from that area.
Okay, so you using the specimen that already been there and just not starting from like scratch. Okay. That's great. The third question is sorry. Have you do a work on a marine fish other than a freshwater fish? That's the question.
Yeah, I mean, the pressure being Indonesian, because people will, why you study freshwater fish as well? You live in the biggest archipelagic country in the world. And even frankly speaking, I don't have a diving license at this point.
You don't have to though.
Yeah, I feel so ashamed.
Move it from the water.
Yeah, but yeah, I extend my wing. So now, like rice fishes that I'm working, I collected a lot from estuarine, which is like the coastal part, like biasanya dibilang air payau, brackish water. And like gobies that now I become more fun to this group, because you can get like 10 species of gobies in one tiny small pond in the east estuarine, or like close to the mangrove. I mean, the more you get to the lower part of a river or like aquatic water close to the freshwater habitats, the more diverse it would be. Yeah.
Thank you. Now the last question, that's actually my question. Sorry. So the question is, how diverse do you think the undescribed, or it's not unidentified, but yang belum dipelajari of our freshwater species? Because I'm interested on doing building a reference library on our like a lake, and then a small pond in Java and Bali. But how do you think the diversity will be? Is it still unexplored, or do you have any suggestions about it?
Yeah, even like southern part of Java, I mean, no scientific papers really focus on the taxonomy. Even now, still there is still undescribed rasbora from Serayu River, for example.
Okay.
And even like how to say it, like in western part of Sumatra, barely any inventory, and like for example in Padang, and even you go further south, there is like a mountain range that encroach the coastal part, and that become like a barrier as well. And just like 20 kilometers away, and you will find different assemblages of fishes. So I mean, I can mention different regions in Sundaland, like in Borneo, that have like so many undescribed species. I mean, like probably we can even find like new genera. Yeah.
Oh, but that's
And also like
interesting.
for example, like pit swarm habitats, rawa gambut.
Yeah.
I mean, that's crazy. I mean, we barely do studies there,
Okay.
but yeah,
That's
you never
yeah,
know.
that's interesting because we are well. I'm interested with more into the environmentology. However, we don't really have the database for the fishes and all our organisms. So we are thinking about what if we are focusing on our lakes because Central Java we have a lot of lakes, we have a lot of apa sih namanya, kolam, and then and we never knew like how's the diversity, but um, honestly, we don't really have a really good connection with the taxonomies. That's why I invite you here because we want people to see this is the real work of the morphologist taxonomist because I'm doing a lot more on molecular. So there will be a great opportunity if maybe we can work on something. We're doing the eDNA for a different from diatom in a couple of lakes in Central Java. So we really want to do a thesis maybe. So I really hope that you can join us to help us describe morphologically the species taxonomy and help me with the phylogenetic as well. So that's the invitation.
Yeah,
Really
sounds
hope,
good.
I really hope that we can find like a small grant to start with that. And by doing that, I hope that we also can bring students with us so they also can learn from directly from the field. So it's not just getting the samples from us the researchers, so they also can improve. So I think that's what I really want to say at the end. But again, I'm so sorry, this is already 10, so I have to close the meeting. Again, Daniel, thank you so much for coming. I'm sure that we will have a chat after this because this is really interesting. For me, everything is interesting, especially something that I don't really know how to do. But just let me know if you are in Indonesia sometime this year or next year, because I really want to let the students see how you do hands-on on how to do the morphology of the species, especially because I just search, of course, that like a very small species, so it's not like a big fish that you've done. So I think that's a lot of work to do that. So, again, thank you so much, Daniel.
Yeah.
Do you want to say something just to close the talk?
Thank you so much also for the opportunity. I mean, it's always a pleasure for me to share something. And then please feel free for everyone. If you are interested, just email me. And even if you are really interested in pursuing further in ichthyology, I would really support you. I mean, I can tell. I mean, I will embrace anyone that really said like genuinely interested, and I can always tell, and people know. I will embrace them.
That's true.
And also about trying to figure like understudied groups. Genetic always like great tools. I mean, I didn't know that the population in South Java is like completely different before genetics. I mean, they look just the same, but then they are sister to something in West Borneo instead of something in Java.
Yeah.
itself. So that's crazy. Yeah, and that's because genetic. Yeah.
Yeah.
Mhm.
Okay. So thank you, Daniel, for a great talk. And I know it
Sama-sama.
is seems long, but again, I learned a lot, and I'm sure I will say this again to my classes in the taxonomy class.
Yeah,
Again, thank
sure.
you. Talk to
Mhm.
you soon.
All right.
I'll maybe just text you or maybe email you again. So thank
Okay.
you again, Daniel. We close the session, and I'll bring back the time to Gaby. Please, Gaby.
Thank you. Thank you, Ms. Dita. Thank you, Mr. Daniel. I believe we are all over here, the participants are all grateful and thankful for the subject for today lectures. Before we do the closing, can we all do a documentation first? So please turn on the camera. So perhaps I can take a screenshot of it, or maybe Pasugi can take the picture and do the screenshot. Okay. Is that all of students? Okay. Okay, maybe no, 1, 2, 3, and then yeah, 1, 2, 3. Okay. Okay. I believe that concludes today's international thematic lecture. Before closing, I would like to close it with a thank you for all the participants, lecturers, including students, Prof. Sabto, and then Sir Daniel, Ma'am Dita, for actually contribute with the best for today's event. Thank you all. I would like to close it. Thank you.
Thank you semuanya. Makasih, Gab.
Thank you, Miss Dita. Thank you,
Terima
sir.
kasih semuanya.
Thank
Mas
you.
Uki, Mas Haikal, semuanya.
Aku izin live ya, Miss.